Okay. So in last session, we looked into a review article, right? We discussed a few key points of that article, and in today's session, we will continue with the topic that we were discussing before that. So I think our last session was on 9th, or sorry, 8th July after the quiz. So during that, after the quiz, we looked into various kind of tissues. So there was epithelial tissues, connective tissue, muscular tissue, and all. So the characteristic features of epithelial tissues we covered already, and we also looked into the what types of muscular tissues are there, and like the skeletal muscle, cardiac muscle, smooth muscles. We already looked into this, and we described the characteristic features of each type of muscle tissue. Let's continue with that. So let's talk about the nervous tissue. So nervous organs basically include the brain, spinal cord, and the nerves, right? So you're already familiar with the structure of the nerve. So these three things together make the nervous organs, and they are responsible for various for controlling the various body functions, right? So they are controlling the body functions. All these organs, they are made up of the main component, that is the nervous tissue, and this nervous tissue is made up of neurons and few supporting cells. So neurons, if you remember the structure of neurons, there is a cell body and there is axon, and from the cell body the dendrites are coming out. So if you recall that structure, we already know that these neurons, they can generate as well as conduct electrical impulses, and this is called action potentials. So basically, from the end of one neuron, means at the uh, synaptic gap, there is a gap between the two neurons. There few neurotransmitters are released at a large amount, and few of these neurotransmitters go ahead and binds with the dendrites of the neuron that is just present subsequent to it, and because of this, an electrical impulse, means there is a exchange of ions that goes down. Sodium and potassium pumps are there that transports the ions into the entire length of axon, and because of which an small electrical impulse is generated. So this is how they pass on the information. Let me just um, yeah. So that is how the neurons, they pass on information, and these neurons are responsible for coordinating the body functions. So in the next point, actually, they have described it. The electrical impulses are received at a neuron cell body. It is also called soma, via the dendrites. And while a single long extension is there, that is called axon. This axon transmits this electrical impulse to the next upcoming neuron. Okay, so this is how the communication between the various nerve cells or neurons takes place. There are various supporting cells. So neurons are the main cells of the nervous tissue, but for the functioning of these neurons, there are few supporting cells are needed, like the astrocytes, okay, and the Schwann cells. Now these cells are not participating in any electrical conduction activity, means they are not involved in generation of these action potentials, but what they do, they help in nourishing these cells, okay, so like say providing nutrients and all. Also they insulate and protect the neuron from the neighboring environment. So this is the role of few of the supporting cells. Now the last type of tissue, so any organ comprised of 5 types. we talked about epithelial tissue, connective, muscular, nervous, and lastly glandular tissues. So these glandular tissues, they are mainly composed of epithelial cells, but these epithelial cells are polarized. Okay, by polarized I mean that they are having a special function of secreting protein products. Okay, so they can secrete protein products, so they are not like the normal epithelial cells that we talked about in the previous class. They are polarized because they are having special feature of secreting some protein products, and these products are secreted directly into their microenvironment. Now, most of the secreted product is made in the RER or the rough endoplasmic reticulum. Okay, and these protein products, usually they are proteins. That's why I'm saying protein products. So they are packaged into small vesicles. That is done by the Golgi apparatus. So if you remember the arrangement of the various cell organelles are surrounding the nucleus, there is endoplasmic reticulum, and there are two types, the RER and smooth endoplasmic reticulum. And you know, RER comprised of ribosomes, which are responsible for secreting protein products. Again, someone is entering. Wait a second. Yeah. So these protein products are packaged into vesicles by Golgi apparatus, and these vesicles are then secreted out by the mechanism called exocytosis. So there are two mechanisms. One is endocytosis, through which cell engulfs any external material into itself, and another process is exocytosis, through which it excretes out. Okay, it excretes out any material into the surrounding microenvironment. Now, these glandular cells are polarized epithelial cells. They have highly developed RER. So these RER, since main role of these cells is secretion, so RER is very well mature, like large. If you see under microscope, the endoplasmic reticulum will look much bigger, like occupying much bigger space in the cytoplasm. Okay, and also the Golgi apparatus is large as well. And you can see secretory vesicles, like empty vesicles, they are floating in the cytoplasm. So these three main components are present, because of which these cells are able to secrete out products, protein products. And like glandular cells, they make up the glands, right? And if we distinguish the different type of glands into two categories, one is called exocrine, another is called endocrine. So the exocrine glands, like the sweat glands which is present in our skin, or the uh, liver or the gallbladder, so these are called exocrine glands. Why? Because they secrete products onto body surfaces, like the sweat. We get sweaty, right? So sweat glands secrete sweat on our surface, skin surface, or they secrete bile juices directly into the body cavities. All right, so that is why they are called exocrine. And these glands can be either unicellular or they can be multicellular. So if they are multicellular, then they will be having some ducts connected to it, through which their products will be transferred, okay, to the desired space, like the skin surface or the cavity, our abdominal cavity there. Okay, there's another type that is called endocrine glands. So these glands secrete the main component, that is the hormones. They secrete hormones, and these hormones are secreted in a very very small quantity. All right, and they are secreted into the bloodstream. So note, exocrine glands don't secrete their products into the bloodstream. They secrete like elsewhere, like on the epithelial surface of the skin, or in the abdomen, right? The small intestine and all. So these cavities they secrete, whereas endocrine glands, they secrete hormones into the blood, and these hormones secreted in very small amount, and they Once we are in the bloodstream, there the flow of the blood helps these hormones to reach their target organ and yeah, start some characteristic signaling process in that particular tissue type or that organ type. All right, so insulin is a hormone, etc. So these are the two types of glands, and these glands are made up of the glandular cells, which are having few characteristics features through which they can secrete protein products. So we saw the two main cell types that are present in almost all organs, the mesenchymal cells and the epithelial cells. Then we saw the tissue types, right? The 5 tissue types that together usually make up an organ, okay? And then an organ is made up of what? Repeating units. These are called functional subunits. So we'll talk about that now. The cells and tissues, they are organized into functional subunits, okay, and each functional subunit is made up of different cell types. What cell types? A functional subunit can be made up of what cell types? Anyone? So if I ask you what all cell types are there in a functional subunit of an organ, can any one of you answer that question? Am I audible? Am I clearly audible to you all? Hello? Am I clearly audible?
Yes, ma'am.
Yeah. So is my question clear to you all? The answer is yeah. Yes,
Yes,
Bhupendra.
yes, ma'am. It's audible.
Yeah, yeah. Am I audible? So I asked you guys that a functional subunit of an organ. Okay. So it is made up of various kind of cell types. So what all cell types are there actually? We have just discussed these. Can any one of you answer what all cell types might be present in a functional subunit? I'm not saying like specific organ, like tell me about liver and all. I'm saying in general.
Epithelial,
Yes, epithelial cells, right? Next, there would be muscular cells, right?
connective tissues like connective cells, blood
Yes,
cells,
yes, correct, correct. What else?
and stated cells, non-stated cells, columnar cells,
Yeah, those are like you're saying
squamous, squamous,
epithelial cells, muscle cells, right? Then you talk about connective tissue cells like blood cells, fibroblasts, etc. Or what else?
squamous epithelial cells, or simple squamous, or complex epithelial cells like this.
So this is subcategories of epithelial tissue in general, like uh, whatever you're describing, like the squamous and the stratified and all. Okay, I'm asking in general, like don't go into subcategories of the tissue types. I'm asking what are 5 cell types, like general cell types you would expect to see in the functional subunit of an organ. See, a functional subunit itself is a mini organ, means it can do all the functions of the organ, since it has to process large amount of say incoming mass. That's why it needs around like thousands of functional subunit together to function together in order to get a large output. But a functional subunit itself can do the same work as the overall organ can. Means if you look at the nephron, which is a functional subunit of the kidney, it can filter the blood, okay, and get the extracted product is urine. But since it is going to since the kidney is going to do the filtration of a large volume of blood, so large number of functional subunit needs to work together, and they all are enclosed in a condensed space, that is your kidney organ. Okay, so whatever cell types or the tissue types that we saw, the 5 types, like the epithelial tissue or the epithelial cells, then connective tissue cells, then muscular tissue cells, right, the skeleton muscles, cardiac muscles, smooth muscles, etc. The nervous tissue, you need the nerve cells to be coordinating the function of each organ. Okay, and lastly the glandular cells. So various cells need to secrete some kind of even the signaling factors and all, okay, in order to communicate among one another. Or if you look at like as I told about exocrine or glands, so there the function is only to secrete. So if you look at overall these five cell types, or the cells that make the 5 tissue types, together make a functional subunit, and a large number of functional subunit make a organ. So this is how you hierarchically understand the structure of our body, overall structure of our body. Am I understandable? Like, is anyone having any doubt on this point? If there's any doubt, you guys let me know. Okay, I'll move. Um, So as I was saying, nephron is a functional subunit of kidney. Similarly, the alveoli is the functional subunit of the lung. And the length scale accordingly varies. Since we are a large, you can say a large bioreactor, okay? So it has large amount of fluids and mass that is coming in and going out. So in order to process all the incoming mass and and generate a large amount of excreting mass as well, the length scale has to be uh, uh, means it is a multi-scale thing. It That is why if you see the organ system, so this is the organ system, the urinary organ system that is responsible for filtering our blood. This organ system, if it is of size say 10 centimeter, then each organ is 1 centimeter, like 10 orders of magnitude less, smaller. Okay, one organ like one kidney is say of 1 centimeter. And if you look into the size of each functional subunit, that is a nephron of a kidney, it is hardly of 100 micron. See the jump or the reduction in the order of magnitude of the sizes of these organs or of these structures, sorry. Okay, so this is how our body is arranged. And when you look at yourself, you are like like very very large in comparison to the size of each of the organ system. So our body is so complex, and when you are going to engineer any particular tissue, you have to first understand what is the size of that tissue. Okay, so and in order to design the size, to decide the size of the tissue, you have to understand the length scales. That is, I'm explaining to you guys. That is why understanding of this length scale is very important. So in the world of tissue engineering, or in the field of tissue engineering, we are hardly able to make 1 centimeter large organ yet. We are still stuck here. We are still stuck here at the 100 micron scale. Okay, so in future we want to go to 1 centimeter scale, and then eventually maybe someday we'll go to 10 centimeter scale of entire organ. We will be able to make the entire organ system. That is how complex our body is. Okay, see here, if you see the size of one cell, it is around, approximately, it is an approximate number, So different cell types have different cell sizes, by the way, but if you make an average of their sizes, it comes up to around 10 to 20 micron. Okay, the size of a cell is 10 micron, and then you saw the size of a functional subunit is 100 micron, 10 times larger, right? And if you do some math, you'll be able to find out that each organ is comprised of 10 to 100 million functional subunits. Okay, not thousands, not lakhs, but millions. 10 to 100 millions functional subunits. So you can easily do the math from here only, from these numbers: 100 micron and 1 centimeter. Okay, so you just have to divide and you'll be able to get the number. So tissue function, this is a very important point that is highlighted in bold, that tissue function is a property of cell-cell interactions. Okay, so one cell cannot function on its own. It needs neighborhood cells for it to pass on some information, like they need to communicate among one another. So how the communication usually happens? It usually happens through ECM, the extracellular matrix, the soluble signals, the cytokines, chemokines, all the growth factors, etc. So they also help in cell-cell communication, and also the cells themselves are, when they are in direct contact with each other through some membrane-bound receptors, the cells can communicate. So when cells communicate, then only they'll be able to, at a larger, like at a when you look at a broader perspective, you'll be able to get the tissue function. Okay, so we will talk about cell communication and all, how each of the three ways, like through ECM, soluble signals, and direct contact, how these three ways of cell communication actually happens. We'll look into more detail, okay, in a subsequent section. But I want you guys to understand that without cell-cell interaction, we won't be able to see the normal tissue function that we usually observe. The cell itself cannot do the complex jobs. Okay, that is why, as I said, 5 tissue types and all, etc. And other than these, there are also surrounding supporting cells are also there. Okay, so tissue engineer usually faces few fundamental challenges when they are trying to make some cell-based therapies. So what are these? First is to reconstruct this complex micro environment that I'm talking about. Okay, in such a way that we are able to see the basic tissue function and the normal in vivo properties in vitro level. Okay, so this is a very complex task, to mimic or to reconstruct the in vivo microenvironment of the cells. Okay, in the in vitro platform, that is in the lab scale, and in such a way that all the function that is usually happens inside the body, that also happens on a petri dish. So it's a very complex job. Now usually people do it, the first step, people do it with few thousands of cells, or say few lakh cells. Okay, and when next challenge is to scale up. Okay, to scale up to say billions, okay, million to billions of cells. That is the next challenge. So while doing it, you need to not create one microenvironment, you need to create billions of microenvironment. Then only you will be able to make a tissue graft that is having, that is going to give some clinical benefit. That's another challenge. Third challenge is to automate the entire system. So every time you are making a tissue, you are getting the exact same properties. Batch to batch variation is not there. So automation is required for that. This is a third challenge. And lastly, implement in a clinical setting. Okay, with all the procedures optimized, like how to handle the cells, how to preserve these tissue grafts and all, all these things need to be optimized when it is actually getting implemented in the clinical setting. So these are the four important steps that a tissue engineer has to keep in mind, has to work, like a ladder, level 1, level 2, level 3, level 4, like this, in order to make a successful tissue engineered product. So this is a very simple diagram that will help you understand how complex the cellular microenvironment is, like in reality. Okay, how complex the cellular microenvironment is. So in the center you will see this is the cell. Okay, this is the cell with cytoplasm and the DNA and all right, and this cell is having some transmembrane receptors. This is the receptors, and these are various kinds of receptors. Few receptors can bind to ECM components that we already know, like the integrin receptors. Few receptors can respond to the mechanical stimuli. All right, few can uh, facilitate direct cell-cell contact. All right, so these are the few receptors for various types of functions. Now surrounding this one cell is of length scale 10 micron, is the fluid. All right, ECM fluid and all. Wait a second. Okay, so as I was saying, surrounding the cell is the ECM, and cell and ECM together make up the tissue. So in the ECM, in between the ECM component also, some kind of fluid is there, that is called interstitial fluid. So the volume of this fluid, or the content in this fluid, gets changed. How? Because of some microcapillaries. These capillaries are very, very thin. Okay, and they are approximately, in a 100 micron range from a particular cell. Means they are at least 100 micron far from a particular cell. So from these capillaries, or the microcapillaries, if they are slightly porous, few of the blood and other plasma material will leak out into the ECM, okay, and make up the interstitial fluid. So that is how, exchange of some components, the fluid components, takes place. Now the content, the blood content that is there in these capillaries, they that gets changed, right? That gets changed. How? So we are moving from the cellular level to slightly organ level. Okay, step by step. So the content of the oxygen, say, in the blood is getting changed, right, because of the working of our lungs. Then if you say the vitamins and all, skin is responsible for secreting vitamin D. Then if you see the how the blood is getting purified, the kidney is responsible for that. And then you need the nutrients, the cells need the nutrients. Then from where the nutrients is coming? So their digestion is happening in our intestine. So that so the soluble food materials like amino acids, fats, etc., they are coming into the blood and through the microcapillaries reaching in the cellular level. So this is how you can visualize, or this sphere helps you visualize the complex microenvironment a single cell is present. Now note these um, small diamond shaped kind of thing. So these are again these are the chemokines or cytokines, soluble factors. Now these soluble factors might be coming from one organ, is present, say, a few centimeters away. That's a very large distance in the body, by the way. Okay, and they are coming in through the blood. Okay, reaching the cellular level to the microcapillaries. So and these factors, they are again very small, like few nanometers of length scale. Okay, and then because of this small size, they're able to bind to the receptors and transmit few signals to the cell, and in response to that, cell makes some changes. Either they change morphologically, or they secrete another new component. All right, or there are some signaling pathways getting triggered. So this kind of uh, changes occur when these kind of soluble factors come in from a very very distant part of our body. So now you are able to understand, like, why construction or the reconstruction of cellular microenvironment is so, difficult in a lab scale. All right, so this peer helps you understand that. Are there any queries till now? Any queries? Everything is clear? Am I audible to you guys?
Yes, ma'am, audible.
Right, so it is clear.
Yes,
Till
ma'am.
now the concepts are clear, so no queries. Okay, thanks. um, now we will move to another section, or the another topic of our entire syllabus, that is tissue dynamics. So till now we studied about tissue organization, how the cells and the tissues and the function subunit or the organ, how they are organized in a very beautiful hierarchical way in our body. Now I will talk about the dynamic nature of the tissue. So under this particular subtopic, we will cover 4 things basically. One is, we'll see what are the dynamic states of a tissue. Then so there are 3 main dynamic states actually. One is tissue homeostasis. So although the word homeostasis corresponds to steady state, all right, so this steady state is not a static one. All right, it changes with time. That is why it's a dynamic steady state. So it takes time to reach a steady state, uh, this time, again that steady state can be something else. So this steady state 1, steady state 2, it can be different with changing time. So tissue homeostasis is, that's why we'll study under dynamics. Then how the tissue is formed is another dynamic state of tissue. It is morphogenesis. It is also called morphogenesis. And lastly, when you're injured, okay, say in internal organ or on the skin or etc., some repair process takes place to healing, to promote healing. So that we will also study under tissue repair. And under tissue repair, we will see few more subtopics, like we will understand what is or how angiogenesis happens, okay, etc. So this is the flow of the topic that we'll discuss under the broad chapter of tissue dynamics. So all tissues are dynamic in nature, all right, or you can say all organs or cells are itself are dynamic. Cells are responding to various stimuli, or they're changing their morphology. So tissue is obviously going to be a dynamic thing. It is not a static. It is not like it is frozen in time. It is changing with time. And each tissue, they have their own characteristic replacement and production rates of cells. So this is actually, you can also term this as turnover rate. Okay, the replacement and production, these two together is called turnover. Means how much of the old stuff is getting replaced by the new stuff. So here it is the cells that are getting made. Okay, so the cells are being replaced by the new cells. So this is the cellular turnover that I'm talking about. And however, the time scales of these cellular processes, they are relatively long. Okay, and one actually tends to overlook their importance. So we will see in this table. So this is a table which highlights the cellular turnover rates in various tissues, not only in human species but also in other species like rat, mouse, etc. So if you see the process of erythropoiesis, which is the process of formation of red blood cells or erythropoietic cells. So this process in rats, it takes only 2.5 days. Okay. Whereas if you see, they have not given for human, right? Fine. So if you see the formation of this small intestine epithelium, okay, so the rate at which it forms in human and rat, it is completely different. In human, it takes at least 4 to 6 days to for entire new epithelial layer to form, and removal of that old epithelial lining, whereas in rat it takes only 1 to 2 days. Okay, then again we can see the difference in the same species and different organ. So if you see in human, the epidermis of the skin, okay, that takes around 7 to 100 days for again to change. Cellular turnover is 7 to 100 days. But in human itself, another type of epithelial, that is corneal epithelial, that is the epithelium present in the cornea of our eye, that takes only 7 days. So my point is 2 points. I'm talking about 2 points. First, same tissue type in different species will have different characteristic time of turnover. Cellular turnover means cellular production and replacement rate. Whereas another point I'm making is that in within the same species like humans, we will see that in different cell type or different tissue type will have different cellular turnover rates. All right, so these 2 points are clear to you all. Any queries? Great, no queries. So so if you see the hepatic cells, that is the liver cells in rat, they can grow. Okay, so you can see that they takes at least 400 to 500 days, like more than a year, for the hepatic cells to in rat to regenerate. Okay, so these are few examples. Now if you look in humans, okay, so this table or this box is talking about the organ types in humans. If you look at the organs, bone marrow, and then the epithelial lining of small intestine and the epidermis of skin, if you look at these three organs and if you arrange it in a decreasing order of proliferation, okay, you will see that the bone marrow is the most prolific organ in our body. Okay, whereas the epidermis is the third most. So these are top three highly prolific organs in our body. The replacement rate of cell numbers in these tissues is on the order of few days. So they take hardly days, like it happens in days, not in months, not in years, but the replacement rate, that is the production of new cells and then removal of the old cells. So that happens in matter of few days. Whereas when you compare this time with other tissue types like say liver, okay, or heart, so for them the rate of replacement is much slower. So as it is stated, cellular content of a liver gets replaced in about 1 year. So it takes 1 year for the liver cells the old liver cells to die and then for new liver cells to take their place. So it takes 1 year, whereas for skin it takes only few days, or for bone marrow again few days. So this is how dynamic the organization or the functioning of our tissues in our body. So as I mentioned in the introduction slide of this chapter, there are three dynamic states of the tissues. One is tissue homeostasis, next the tissue repair, and third is tissue formation or morphogenesis. So briefly in 1-2 line, I have described these three processes, but but what we'll do, we will study each of these dynamic states of tissue in detail, one by one. Okay, as as a subtopic. So tissue homeostasis basically refers to the normal steady state function of a tissue. And again, let me repeat, the steady state can be multiple, like a tissue can be present in multiple types of steady state. So normal, like when it is healthy, it is functioning in steady state 1. Now suppose it is injured, then steady state is changed. Okay, that then the steady state steady state 2 is there. Or suppose it is undergoing replacement, as I said, replacements and all, the new tissues are formed, new cells are forming, old cells are dying. So that is again steady state 3. So if it is secreting something, steady state 4. So with time you will be able to observe that steady states of a particular tissue is changing. But we define steady state for a very long period of time. Like other than bone marrow or epithelial lining of the small cells, like where the replacement rate is in order of few days, if you look at heart, you will see that heart is present in one steady state for years. Okay, so then you feel like it is static, वही तो काम कर रहा है. Okay, but steady state can change a little bit. So tissue homeostasis basically refers to the normal steady state functioning of a particular tissue or an organ. So as I was saying, the bone marrow and the skin, if you look at any time point, you will see that these organs are producing cells only. Okay, the major for the major time they are producing cells. Then if you look at the glands, like the endocrine glands or the exocrine glands, they are producing protein products or secreting some protein products. Or if you look at the kidney or the lungs, they are carrying out mass transfer operations. Means some product is coming in, they are performing, so for lungs filtration and all, exchange is happening, right? Exchange of gas is happening. For kidney, filtration is happening. This is a mass transfer operation, by the way. And then the whatever product is there, it is going out. So this process is happening steadily. Like if you look for heart, it is constantly pumping blood, right? It is acting as a pump, right? That is a steady state function. And then when you look at the liver, it is acting as a biochemical refineries. You know, liver is a very important gland in our body, where all the important biochemical reactions like fat synthesis, protein synthesis, carbohydrate synthesis, or if you say the drug metabolism, everything, it happens in the liver. So the metabolic pathways are multiple metabolic pathways are occurring simultaneously, one by one, and some input reactants are there, and you are getting some products. So that's why it is called biochemical refinery. So this is this normal steady state function. Okay, where tissue repair, this tissue repair state, the dynamic state of tissue repair only happens, it does not happen in normal state. Okay, tissue repair only happens when you're injured, when the organ in question is injured. At that point only, the entire tissue repair mechanism, okay, it's a separate mechanism altogether. It's completely different from the normal functioning of the organ. It starts, this mechanism starts. And this is very important for us to understand, because why would anyone want to surgically implant a tissue injured product? Why would they want that? Because they are surgeon is looking for repairing. Surgery is looking for repairing a particular deformed tissue. So, which is why it is very important for us to understand the entire tissue repair mechanism, all the steps that are there, what all cells are involved, how the levels of various biochemicals are changing during the entire process. It is important for us to understand. Then only we can ensure successful grafting of a particular tissue engineered medical product. And the last dynamic state is the formation, that is morphogenesis, tissue morphogenesis. So the knowledge of this area is coming from the broad subject of developmental biology, okay, and it is again important for us to understand, because if we don't, then we won't be able to completely mimic the tissue in vivo tissue microenvironment. All right, we will look into this as well in detail. So today we will finish the broad topic of tissue homeostasis by looking into the few examples of only the prolific tissues, the highly prolific tissues in our body, which are bone marrow, the epithelial lining of small intestine, and the skin. So we will look how these three organs functions. So for the bone marrow, we already looked into it, like how many cells are there, how many days it takes for the entire cell content to be replaced in the first in the introduction part, I think. So for bone marrow, only thing I have to say is that it is the most prolific organ in our body, and the entire cellular content of bone marrow, like I think it contains around 400 to 500 billions of cells. So all these cells, they get completely replenished in 2 to 3 days. Okay, that is why it is a highly prolific organ, and that is why the bone marrow transplantation procedure is very, very successful. So I think for some of you guys are already familiar with the terms to stem cells. Okay, so these prolific tissues are able to produce this much amount of cells at a very high rate because they contain stem cells. Okay, because they contain stem cells. These stem cells are able, they are not mature cells, they are able to proliferate, and each of the daughter cells then undergoes differentiation and form the mature cells that is there, that you see in that particular tissue. So because of these stem cells, these tissues are having this prolific nature. Next, we'll look into the how the small intestine's epithelial lining is maintained. Okay, how means the cells are getting proliferated and then differentiated. So if you look at the structure, of the small intestine epithelial lining, you will see that there is small finger-like projections which are called villi. So in this figure, you will see that this is the surface of the small intestine epithelial lining, and there you will see the villi, and each of these villi is also having the trough region, the valley types of region, that is called crypt. So the crypt and the villi is plural, singular is villus. So these are together making the entire surface, the top surface, the surface of the epithelial lining of small intestine. Now this entire surface is highly dynamic in nature. All right, is highly dynamic in nature because the cellular content turns over in approximately 5 days. Like every 5 days, the entire small intestinal lining gets changed. That is why it is the second most prolific tissue in our body. So how actually cells travel? Let's look into that. Okay, how the cells are moving up and then forming the layer, then going down, dying, like how it is actually happening. So these are the few points that are highlighting that, and I'll also explain it using the figure that I showed. So towards the bottom of the crypt, so crypt, I mean the troughs, the valley regions in the on the surface of the epithelial lining. These at the bottom of these crypts, there are stem cells. Okay, so these are tissue specific stem cells, means you will find these stem cells particularly in these small epithelial linings. Okay, you will only you will find here only, will not find these stem cells anywhere else. And these stem cells, they can they are very slow dividing. Why? We will see later that the stem cells, the stem cells are present in a very small number, and they only start dividing when it is needed. Otherwise the entire reservoir of stem cell will get finished up, right? So we don't want that. So the organ also doesn't want that. So these are, because of which the metabolism of these cells are very slow. And in each crypt, or at the bottom of each crypt, there are approximately 20 of these only. Okay, say, think like only 20 are there, and from the 20 you will get millions of cells. That is how rapid that cell division happens. After division, so say among 20, one stem cell get activated. Okay, and starts dividing. It will divide and make a large number of daughter cells. Now each of these daughter cells, they will either two things can happen. One daughter cell again becomes a stem cell and fill up the reservoir, or both of the daughter cells will now go towards the differentiation process, or one daughter cells remain become a stem cell, and other daughter cell becomes differentiated cell. So this kind of stochastic, in this stochastic way, the cells can decide what to do. So say in this example, say both the daughter cells decide that they will form the differentiated cell. So the daughter cells then move up the crypt. So division happens at the bottom of the crypt, that is the trough region, and it starts to move up. And as it is moving up, it is, again kind of dividing. Okay, so it can be in a stage that is called progenitor cell stage. So stem cell and progenitor cells, these are basically one after the other. Okay, they like if you say like this is the stem cell, it divides. Okay, daughter cells are formed. Now one of the daughter cell decided to become a progenitor cell. So what is the difference between a stem cell and a progenitor cell? So progenitor cell is usually unipotent. Okay, it can only create one type of cell, one cell type, whereas the stem cell, if given the right environment, it can differentiate into multiple cell type, or it can give rise to multiple cell type. So in order to give rise to millions of cells, we need progenitor cells. Okay, they will start dividing, and each of these progenitor cells will then start to differentiate. Now as this division and differentiation process is happening, the cell is moving up the crypt. Okay, so once they have left the crypt, okay, the trough region, they are fully mature. They now enter the base of the villi. So have a look at this diagram. This is one crypt, and they have zoomed in into what is happening at this crypt. So at the bottom, here at the bottom is the reservoir, stem cell reservoir. They are non-dividing. They are actually having a very slow metabolism. Now say one of the stem cells get activated and divided and gave rise to the daughter cells. Now one of the daughter cell can be a progenitor cell, one of the daughter cell can be stem cell, or both can be progenitor cell, something like that. So if they are progenitor cell, so they will start to divide and differentiate. Now as it is happening, they will move up the crypt. Now this is the top of the crypt. Like at this region, all the progenitor cells have divided. The reservoir of progenitor cells, you can say, has finished. They have changed their morphological characteristics into mature cells or differentiated cells. Now this region and this region is same. So this is basically the base of the villi. So at the base of the villi, we will find the mature epithelial cells present. Now what happens? Now these mature epithelial cells, which are there at the base of the villi, they move up the villi. They have to climb, and it happens over a period of 5 days. Okay, and once they reach the top, okay, the top of the villi, the cells at the top, they starts dying and they shed off. Slug off means shed off. Now while this is happening, okay, now sorry right, so this is one crypt, this is one crypt. From both the ends the cells are climbing, and at the top there will be no cell division, rather this layer gets slugged off, like it sheds off entirely. Okay, then again new cells will climb off, then again they shed off. This is how the movement of the cell is happening, and while they are moving, they're also changing their states, like from non-differentiated state to differentiated state. Not only they are changing the states, they're also doing the important function. What function? The function of absorption of nutrients and digestion of nutrients, like they are also performing that function as well. So you can understand how complex it is. First you have to mimic the dynamic cellular differentiation states that the cells are in. Then you have to also make sure that these cells are doing the functions of digestion of nutrients and absorption of nutrients. That is the main role of small intestine, as you guys know. So that is also happening at the same time. Now this is all about how the homeostatic state or the normal steady state of the small intestine, of the epithelial lining of the small intestine is maintained, right? Now we'll talk about the same thing for skin. So skin has two primary cellular layers. One is called the dermis layer, another is called the epidermis layer. Okay, so dermis layer is basically the connective tissue layer, and so these are, this is epidermis. Say this is epidermis. There is one layer that it is not a cellular layer. It is called a basement membrane or basal lamina, and below basal lamina is the dermis layer. So this is cellular layer, this is cellular layer, and in between the two cellular layer there is a ECM layer. Basically it only composed of ECM components like collagen, etc., and it is the basement membrane. So below the basement membrane is the dermis layer, and above the basement membrane is the epidermis layer. So dermis mainly is composed of the cells, that is fibroblast, but there are other cell types too. All right, but I'm talking about 90% of the cells are fibroblast. And then the basement membrane, if you go up like this, then you will be understand the points. Now the basement membrane or the basal lamina is composed of ECM components, and it majorly collagen type 7 is present. And on the outside of the basement membrane, that is at the top of the basement membrane, is the epidermis, and it is again majorly composed of the cells that is called keratinocytes. So these keratinocyte cells are like the small intestinal epithelial cells. How? They're also under a different differentiating states. Okay, so we'll look into those differentiating states. So because of these different differentiating states of the keratinocytes, The epidermis can again be divided into 4-5 layers itself. Now, epidermis is a layer structure, as I already mentioned, and the cells that are located near the basement membrane. So there, the cells with the least replicative, that is this layer, the layer of the cell that is present near the basement membrane. There, people have found few stem cells are also there. Because of this presence of these stem cells, say few, these are dormant cells, as I already told. One of these stem cells gets, say, activated and starts dividing. So now these divided cells, they will undergo differentiation. Now the entire differentiation process happens layer by layer, and as the cells are getting differentiated, the cells are moving up. Moving up means moving up from the basement membrane to the surface of the skin, where the least differentiated cells are there at the basement membrane or near the basement membrane, and the most differentiated cells are there at the surface of the skin. So this point will make my point much more clearer to you guys. So as the cells leave the basement membrane or the basal lamina, they undergo differentiation to first prickle cells. So here is the diagram. This is the connective tissue. This is the basal lamina, just above the connective tissue, that is the dermis. Just above the dermis, the basal lamina is there. Now the cell, that is the cellular layer that is just in contact with the basal lamina, is called basal cell layer, or it is also called stratum bacilli. Now here few of the cells are stem cell, means they are stem cell in nature, this having the characteristics of stem cells. Now one of the cells starts dividing, and the daughter cells now goes through different stages of differentiation. First, the cell become a prickle cell and give rise to a layer called prickle cell layer. Okay. Next, few cells, then they move up. Now, why the cells are moving up? You might think. Okay. It is because the surface of the body surface is limited. Okay, there is no way the cells can move in xy direction. It cannot move in planar direction. If the cells are dividing, it has to move up, like in z direction it has to move. Okay, our body surface is limited. Now the prickle cells, the cells in the prickle cell layer, they are then moving up and becoming granular cells, are forming the granular cell layer. Okay, and then few of the cells, or the top of cells that are present at the top of the granular cell layer, they starts dying, they starts dying and become extremely keratinized. Means keratin is a protein, and in these cells the protein content, the keratin content in these cells is very high, so that is why they are called keratinized cells. So, and also they are dead cells, and they form a very thick layer that you actually see, that is actually visible to you. This is the layer that is visible to you. It is called keratinized squamous. By squamous, like they are having a flat kind of a morphology, that's why it is written squamous. And this layer is quite thick. It's like 20-30 layers thick, this layer. And as I already told, that we shed our skin, right? I've mentioned previously, the top layer of the skin, it flakes off. It flakes off like in once a month, you can say. We shed our skin once a month, so that the new cells that are present just immediately below the shedded off skin gets exposed. Okay, and in this way the cells are moving up and then replenishing the shed off layer. All right. So the prickle cells that are there in the prickle cell layer, it is also called stratum spinosum. All right. Then this prickle cell become granular cells and form the granular cell layer, or it is also called stratum granulosum. Okay. And then the cells become keratinized, all right, and become flat and form the stratum corneum layer. So this is a dead cell layer. So after then few days, like once a month, these cells, the cells that are present at the top of the stratum corneum, they flake off from the body. So one should note that the cells that are present in the basal lamina or on the top of the basal lamina, that is the cells in the basal cellular layer, only those cells are cycling, means only those cells have the potential to divide, whereas the other cells present at the top of the basal cell layer, stratum basilae, they are differentiating. They are not dividing, they are differentiating. Now it is believed that 1 in 10 to 12 basal cell layer is a progenitor cell, all right, which can only give rise to one type of cell. And because of this cell, we are able to get large number of cells that are present in the top layers of the epidermis. Now net proliferative rate of the skin varies with the region of the body. Okay, means say at the palm or the sole of your feet will have a different proliferative rate in comparison to your face or the lips, right? And the turnover of the skin means the cell production and cell replacement rate. This rate is in the order of few weeks, and because of which a large amount of flakes is generated over time. So whatever dust that you guys see in your home when you are using broom to clean, most of the dust is actually the skin that we have shed. Okay, the house dust is coming from that only. So this is how the skin layer or the epidermis layer is undergoing a continuous differentiation process, or it is always present in some differentiation state. The different layers of the cell are present in some differentiation state, and the top layer gets flaked off, and the in the bottom layer, that is the basal cell layer, is responsible for generating new cells. So any queries till now about how?
Yes, ma'am.
Yes. Yeah.
Ma'am, actually, I have two queries. Like in skin, that when a burn case is caused, then the another part skin is attached to the affected area. So in that skin they are putting on that affected area, they also have that stemness property like that. And one more question is that when a baby is born, her
Hmm.
size is small.
Hmm.
Her skin and epidermal layer is also very small. When the
Hmm.
baby is growing up, that the cells are also dividing
In,
and size is also
increasing.
increasing. Yes.
Correct.
So when the precursor layer is dividing
Hmm.
and in the horizontal way,
Hmm.
how how it can be maintained that it didn't go to the granular level cell layer, because after dividing it should be go to the granular layer.
Acha, okay. So I'll answer your second question first. So you're talking like when a child is growing up, the surface area of the body is increasing, right? So the cell number is increasing in the XY direction, and you're asking how in the granular layer or in the Z direction how the cells number is increasing then, right? That is the second question, right?
Yes, ma'am. And how they maintain the precursor cell line maintained and didn't go to the granular layer.
I didn't get it. Means why it won't go to granular layer?
Because then after the cell divide, that they should be go to the granular layer. After that, the keratinized layer, na?
Yes,
So
yes.
that
One
the
by
skin.
yeah. So cell division will only happen at the basal cell layer. Okay. Other than that, in no other layers cell division will happen. All right. So when a child is growing up, at that time, the cell division is like cell morphology is also changing. By the way, okay, the number of cell layers is also changing. You'll see, a baby will have a very thin skin, whereas an adult will have a thicker skin. So environmental conditions, etc., dictate this kind of things, also your genes and all. So the division is happening in both directions. It is logical. Otherwise, how will you explain how the cell is dividing? and and also the cycling of the means this process, this flaking of process is also happening simultaneously. So we are talking about billions of cells are getting produced throughout the body. All right. So I think if you imagine in this way, you'll be able to understand your question, like it will be clear to you only the question that you asked. Okay. The cells will grow. It will not, like the cells will not go to glandular layer. Cells will go up in z direction as well. That process will happen. All right. In addition to the cell division that is happening in the xy direction. Now Bhupendra, your first question that you said about the burn victims, right? Can you repeat that question again?
I've said that when the burn caused that the reddish layer, like the connective tissue layer, should be exposed, and after the treatment, another place of the unaffected area, the skin was peeled off and apply on the affected area of the same person.
Hmm.
So that applied skins have also have that property of stemness, or they have lost it.
So we have not yet covered the tissue repair process, but I can see burn. The level of burn, it can be such that it only destroys the epidermis layer. It can happen, or it destroys both the dermis layer as well as the epidermis layer. So what people usually do, for if the victim is not having the epidermis layer, then there is a process, in vitro process, in that people can do in lab scale, through which you can generate this kind of a layered epidermis structure. So they do that on a particular substrate, say in hydrogel and something like that. And when it is done, see, skin cells very well divides in vitro. So it is the cell, increasing the cell number is not a problem in that way. That is why this process of cell differentiation in the z direction of the epidermis is possible. So once they have done it on an artificial substrate, say hydrogel polymer, etc., they take that and suture it on top of the exposed dermis layer. Now, if and now you ask that the stem characteristics. So usually what people expect, the wound healing process to begin first, because they want that area to recover. It should not be having inflammation or infection, etc., like that. So their primary concern is this. Later on, what is the expectation is the surrounding cells, the wound area is having some surrounding cells, right, at the periphery. It is expected that the host cells will start to migrate, okay, and will replace the cells that is transplanted and take over and form a normal tissue. So when that happens, then the stem cells population that is there in the basal layer, which is then the surrounding tissue, they will also start migrating. So the effect of the tissue graft that we gave is temporary. All right, keep that in mind. It is only temporary. Its role is to heal. Okay, its role is to not provide stem cells. And in the second scenario, when the dermis layer is also injured, then the problem becomes a bit more tricky, because the dermis layer is the layer which is having the network of blood vessels. Then they have to engineer in such a way that as soon as possible a network of blood vessels gets established at the wound area. All right, so it becomes tricky, more tricky actually. The epidermis layer is not having any blood vessels. uh, Means the basal cell layer, whatever nutrients it wants, it gets it from the capillaries that are there at the dermis layer. whereas the above differentiating cells, they are they do not have that much nutrient requirement. Okay, because they are not dividing, they are differentiating, so there's a difference. So uh, is your question clear, or you have any follow-up query?
No, I'm clear, ma'am.
All right, anyone
Thank
else having any queries related to this topic, or anything that we have covered today or in previous classes, anything you guys want to ask? Okay, no queries, so I'll take the attendance, and then today's session will be over. Okay. Stop recording.